This is part 2 of the genetic summary video. You've dealt with Mendel and his laws all the way up to sex determination, sex-linked traits, and we carry on with number 6, blood types and compatibility. The blood type name comes from the type of antigen present in the red blood cells. So blood type A cleverly has A antigens, B will have B antigens, AB has both A and B antigens, and type O has none. And antigens are things on the surface of red blood cells that may trigger a response in a receiver, in the receiver's immune system. Now, if we have A antigens, it means we have B antibodies. Type B blood has B antigens, and therefore it'll have A antibodies. Type AB has both antigens, so its antibodies it will have neither. And type O has no antigens, so it'll have both A and B antibodies. And antibodies can have a variety of functions, but one of them is they're like a first alert system. Often they will tell your body that you've been invaded by a foreign thing. In this case, it might be a wrong type of blood, but it could be by a virus or bacteria or something like that, and they trigger an immune response. In a blood transfusion, what we have to do is avoid having an antigen be identical to an antibody, because if we say had A and A get together, A antigen with A antibody, we're going to have agglutination, and that's the clumping of the blood and it won't flow well, and that's a problem. If we look at type O is a universal donor. It has no antigens. If it has no antigens, it doesn't matter what antibodies are present, you're not going to get agglutination. And type AB is called the universal receiver. It has no antibodies, so there's nothing there waiting to pounce on any of these antigens if they're present, so it can accept all their blood types. But let's take a look at blood type A. What can it be donated to? Well, blood type A has A antigens, so it can be donated to blood type A because it has B antibodies, not a match there. Can't be donated to B. It can be donated to AB because there's no antibodies, and it cannot be donated to type O because we see an A here and an A there. They would get together and agglutinate. On the other hand, what type of blood can type A receive? Well, it has B antibodies, so it can't receive type B or AB because those would match up and you'd get agglutination. It can receive A, and it can receive O because we don't get a match to the B antibodies. So that's looking at the types and the compatibilities, and you have the full chart in your notes. Blood type genetics is relatively straightforward. We look at this, it's an example of what are called multiple alleles, meaning we have more than 2 alleles. We have an allele for the A, allele for the B, and we have a recessive gene symbolized by a small I. But you only get 2 genes for blood type, one from mother, one from father. And that means turns out there are 6 combinations. Now, A and B are codominant, and that means when they're both present, both traits are expressed. And we look at what the results are. We work backwards here. We look at the phenotype, and we go back to what are the genotypes. Type A has to have the A gene, and then it really doesn't matter what the other gene is unless it was a B. So in this case, it's homozygous dominant. In this case, it's heterozygous. Both code for A. And the same thing for B. We have 2 genotypes for blood type B. For AB, we have to have the A gene and the B gene. And for type O, we have the 2 little I's. We can't have an A or a B present. So those are the blood types. And when you're doing questions, you want to know the difference between homozygous and heterozygous. And you have to usually work out these genotypes before you load them in a Punnett square. Once you've done the Punnett square, you look at genotypes and you go back to working out the phenotypes. Test cross we did before spring break, and this is where we're trying to determine the genotype of an organism that has a dominant phenotype. If it's a recessive phenotype, we know what the genotype is. It's got to be homozygous recessive. But if it's dominant trait, then we have to work out is it homozygous or heterozygous. So the procedure is to mate the organism with one that has a recessive trait, and you'll look at the offspring. Now, anytime you get the recessive trait appearing, there's no doubt about it. The organism with the dominant phenotype has to have been heterozygous because it has to have had the recessive gene that it could donate to the offspring and get together with the other recessive gene. There's no possibility if it's homozygous dominant, you'd get a recessive trait. On the other hand, if all you get is a dominant trait, it's probably going to be homozygous dominant, but you never know for sure because both homozygous dominant and heterozygous can produce a dominant trait. But the more times you do it, you increase the likelihood that it was homozygous dominant. Incomplete dominance, codominance, we talked about that earlier. And incomplete dominance means when you put two genes together, in this case you don't get one trait or the other, one doesn't dominate over the other, you get a blending. So if we had the white rose with the red rose, we'd get a pink rose if we had both genes present, and that's a blending. Codominance, that was like where we had where we talked about the cows and you had the red hair and the white hair, and if you have both genes present, you get both red and white, and cleverly that's called roan. Dihybrid crosses, best to go right back to those notes. Get in the habit of figuring out how do those genes combine so you can load them across the top of your Punnett square and down the left of your Punnett square. And remember, they're going to come in little pairs on the outside, and in the 16 boxes, you're always going to end up with 4 letters per box. And then you just have to interpret them for each of the 2 traits within it. Now this year, you are not getting ready for a genetics test, so you're really trying to use a summary to help you get through some of the worksheets. And some of you are already doing this. You're asking questions and things like that, and that's great. But keep trying questions, make your best guesses, ask for help if you need it, and do your best.